It's a little after 7, so everybody take a seat and quiet down. What was the word of the day?
Methylation.
Methylation. I guess you'll say that many times during your talk.
Probably. That's what we do.
I don't have too much to announce other than I'm here on short sleep. I got home from Boston last night at 3:15 in the morning on a late flight. But it was a nice trip to your old stomping grounds there. So today's talk is by J. Patel, one of our first-year fellows, Epigenetics Explained. We got a couple of people over in Israel. I don't know if they're listening to us. There's a meeting going on. Steve Tillis is there. And if you guys have nothing better to do and you're listening into Seattle, send me a text message. All right, Jay.
Alrighty, so today my topic is epigenetics, and when I start to first research the topic, I quickly learned how big of a topic it is, how much information there is out there, and how much we still don't know. So that'll kind of be the theme, and how everything is related that we talk about. So everything I talk about is might seem like one specific process, but it's generally never in isolation, and there's no absolutes. So, and then there's been many new developments in the last 5 to 10 years, some pertinent to our field, specifically observations in asthmatic patients and the Th2 immune response. And then more recently, it's been helpful to understand this in the context of tumor biology and therapeutics for cancers and hematologic malignancies. However, it's still kind of up and coming and in our field. And I have no disclosures. So first, as an introduction, I wanted to just ask the following questions. What is genetics? What is epigenetics, and why it's important? Because you have to really understand what genetics is to understand what epigenetics is. So genetics is the study of the genetic code, specifically DNA, and also includes its variation and how it's inherited. And we know that genetics cannot explain everything, specifically monozygotic discordant twin studies, how environment can affect the development of different phenotypes of monozygotic twins. And also, pertinent to us is the rapidly increasing rates of atopic disease and asthma, faster than the genetic change and variation. So first I wanted to talk about DNA and the organization of DNA. So DNA is organized into chromosomes that are generally in the nucleus, and the reason, and then understanding why DNA is organized the way it is. So it's tightly wrapped around structural proteins called histones, and it forms a complex called a nucleosome, and that has a couple hundred base pairs wrapped around the histone proteins, and then those nucleosomes are tightly bound together to form chromatin, and there's all this tight compaction to fit the vast amounts of genetic material in our chromosomes. There are 8 different histone proteins, and then the H1 histone protein sits atop an DNA and histone protein complex, and we'll talk a little bit about that later. So it kind of leads to the following questions. How do cells with the same genetic material differentiate into different cells with different function? We have our code to pretty much for gene expression for many different things, but however, not all our cells express the same genes. How does the environment impact that genetic code? And again, that question about the discordancy in monozygotic twins. And then the increased incidence of asthma and atopic disease. And the answer, in part, is epigenetics. And these are just a bunch of words to kind of describe epigenetics, related to epigenetics, and then its use. So going forward, the rest of my talk today, we'll just define epigenetics, look at specific mechanisms needed to understand the principles of epigenetics, review some of the tools that are used to study epigenetics and the epigenetic code, and look at the epigenetics and the role in asthma, atopic disease, and other things that we have a little bit more information about, like exon activation, cancer malignancies, and then just data about how environmental influences affect that. So epigenetics, what it literally means is, above the genetic code, outside the genetic code. So it's everything but, the genetic code. And it's not dependent on the DNA sequence specifically. Now the different mechanisms that we'll talk about, these are the more common ones, and there's more than this, but these are kind of the principal ones to understand, and some of the ones we have the most information about and are the most well understood, and that's DNA methylation, histone modifications, and then non-coding RNAs. What's not in here is chromatin remodeling, which kind of goes along with histone modifications as a part of chromatin remodeling. And all these mechanisms are involved in gene expression, and it affects the DNA's interaction with different transcription factors, transcription stability, DNA folding, nucleosome positioning, and chromatin compaction, as we talked about earlier, how that DNA is organized. So first, I was going to talk about DNA methylation. This is the first epigenetic mechanism that was initially described, and it's the most well-studied. We have the most information about this mechanism, and what it is a covalent modification on a cytosine to where a methyl group is bound to the cytosine, and it's mediated by different methyl transferases. The three most common ones that I'll talk about are DNMT, or DNA methyl transferase 3A, 3B, and 1. And it's important to sort this out from histone methylation, because that will is mediated by a different enzyme, and that will have different effects than DNA methylation. And it's important to understand that each cell type has unique methylation patterns. From during embryogenesis, initially there's a period where nothing really is methylated, and then subsequent cells, as they become more and more differentiated, have more or different methylation patterns, and specifically very important in hematopoiesis and maintaining stem cells.
Since you mentioned embryogenesis, I've always thought of this as a process that happens like after you're fully formed and then you're exposed to cigarette smoke and environmental factors. But this occurs in embryogenesis very early?
Yeah, so there's information on not just cigarette smoke, but pollution. There's just so many different environmental factors like maternal diet, the microbial environment, and really how that has changed in the last 20 or 30 years now, or even greater than that, as we've become more of an industrialized society, and how that's different than some of the what's in the environment in the like the farming environment, and how that's very key in kind of understanding the development of some of those diseases. So first, in terms of this, is a picture to show what's going on with DNA methylation. So you have the DNA methyltransferase enzyme that uses a methyl donor to catalyze a reaction where there's a covalent bond on the cytosine, and it becomes 5-methylcytosine. And that's, in general, for the most part, cytosine is the primary nucleotide that is methylated. And we'll talk about later, but it usually occurs in context of groups of the cytosine and guanine nucleotides called the CpG, which it stands for cytosine, a phosphate group, and then guanine. And this is more of a permanent, I'll say relatively permanent, because, like I said, nothing is really absolute in epigenetics, but it's a more permanent sort of modification. But it can be reversed and changed. So DNA methyltransferase 3A and 3B are primarily responsible for de novo methylation. So methylation that occurs in cells in the sequence that isn't previously methylated. And it does methylate DNA that already is partially methylated, like during the replication. You'll get 2 different strands of DNA, that half of them which is methylated from the parent, and then the new strand isn't methylated yet. So it does also act on these on the new strand as well. But the importance is the rate of the enzyme is similar to the methylation on these new the unmethylated DNA strands versus the DNA strands that are half or hemimethylated. And loss or changes in this in the enzyme or enzyme function can really affect expression of genes, especially in gene silencing. When you inherit 2 different chromosomes, there might be silencing of the maternal or the paternal genes. So alterations in these enzymes or this process can affect that. And then the DNA methyltransferase 1 is the most abundant DNA methyltransferase. So this one is its primary role is to act to maintain methylation in replicating cells. So, and then the activity is much increased on the strands of DNA that are all already methylated. So this is versus the DNA methyltransferase 3. And this picture it shows the overall process of hematopoiesis and the role of DNA methyltransferase in that process. So it's a pretty complex process here, but the important thing to kind of take away from this slide is it's such a balance of methyltransferase activity in renewing a stem cell and then also pushing it into different forms of differentiation. So the CpG Islands, so like I said earlier, is the covalently modified cytosine. So it generally is with cytosine and guanine. So these are, methylation usually occurs at parts of DNA that have increased amounts of cytosine and guanine sequences. And they're usually located in regulatory regions of the promoter enhancer regions of transcription. And generally, it's the role plays a role in silencing of genes. However, again, like I said, nothing is completely absolute in epigenetics. And the CpG islands are generally at least 200 base pairs, and more than half of those base pairs on the CpG islands have cytosine and guanine. And they're more differentiated and they're more methylated and terminally differentiated cells versus undifferentiated cells generally have less methylation. And the other kind of key thing with CpG islands is there's a lot of palindromic sequences, so it's meaning that it's read same front as, or like 3' to 5' as it is read 5' to 3' to help kind of maintain that methylation sequence. So the mechanism of gene silencing, we're still learning more and more about it. It's not a process that's completely understood. It has been shown to limit access of transcription factors by methylation, and it's not just acting alone. So there's definitely the recruitment of other histone enzymes and other epigenetic modifications, and how much of the gene silencing versus activation is purely the methylation versus the other interacting factors in epigenetics. So this is kind of a replication fork. So this is a illustration of what I was talking about earlier about how when DNA undergoes replication, there are methylated cytosine that, so on the new strands of DNA, initially they're unmethylated, and then DNMT1, or the DNA methyltransferase 1, acts pretty closely with the replication fork to help preserve that methylation in replicating cells.
Use the pointer.
Oh, yeah. Right here. So, as I discussed a little bit earlier, so it plays a key role in imprinting genes, so that there's silencing of certain genes that are more that have two alleles. So certain genes will be more maternally expressed and others paternally expressed. And it's not just imprinting; it's not just DNA methylation that plays a role, but it's also histone interactions, modifications, and other non-interfering RNAs that are all part of that process. So one of the key places where methylation is very important is during inactivation. So what happens in inactivation is that the paternal X chromosome is initially inactivated during embryogenesis, and then after a few divisions, it gets reactivated, and then there's a random inactivation of either the maternal or paternal X chromosome, and the methylation is responsible as one of those mechanisms. However, there's other epigenetic mechanisms as well, and its disruption can result in certain autoimmune diseases that are seen, such as lupus, autoimmune thyroid disease, scleroderma, that are localized to the X chromosome.
That's interesting. Do they know what? So there must be a lot of genes on the X chromosome specifically implicated in those autoimmune diseases. Is that well known, or did he come across it?
I remember reading about it. I mean, I think they've localized it to the X chromosome in terms of, I think they've looked at patients with lupus and thyroid disease, and I believe they've looked at methylation patterns and stuff and seen differences. So I think a lot of these mechanisms aren't clearly elucidated, but I think in a lot of what I was reading, there's patterns they're seeing, as that kind of goes with a lot of epigenetics, and they kind of make conclusions of, is the pattern that we're seeing kind of clinically relevant? So,
I mean, it's an interesting question because, as I remember, autoimmune diseases are about tenfold more common in females than males. Does this somehow explain it if we take this to just clinical medicine?
I mean, yeah, because in males you have only one X chromosome and it's not inactivated, so it doesn't explain everything because males do get these autoimmune diseases as well.
Yeah.
But yeah, I think it definitely there is a role for that. Yeah. So this is just looking at the embryogenesis and X inactivation, and there's a lot of kind of text at the bottom, and all of that is just all the different roles of epigenetics in this process. So like I said, it's not just DNA methylation, and it's other histone modifications and other things as well. And the X is...
Use the pointer.
Oh yeah. So when it talks about this paternal gene that, protein that is responsible for this process. So this is acting through methylation up here, but then here there's other methylation and histone modifications that are playing a role in this as well. And then, so now I was going to start to talk about some of the histone modifications. Histone modifications, to understand that, it's kind of going back to taking a look at the bigger picture of euchromatin and heterochromatin. So euchromatin is more transcriptionally active. It's less condensed, it's more open. There's more room for transcription factors to access the genome for transcription. Heterochromatin is more densely packed and is transcriptionally generally inactive. And there's a large amount of influence from histone modifications, proteins, histone protein modifications, and then also chromatin remodeling complexes, which are other proteins that come in that are also play a role with it, in terms of changing up the nucleosome. So like moving around the histones physically, and we'll just kind of open up the DNA for transcription. So the histone octamer first is organized into, like I said earlier, two different. There's 8 different histone proteins. There are 4. There are 4 of them, and then that are duplicated. So there's two H2As, 2 H2Bs, 2 H3s, and 2 H4s. So that's how it forms an octamer. And when the DNA is wrapped around that, a couple hundred base pairs, it's called a nucleosome. And there are N-terminal tails that protrude from, which are the amino acids protruding from the nucleosome that are attached to the histone proteins. And that's the location of the majority amount of histone modifications. So, in terms of naming the different types of histone modifications, you generally take the histone type and then use the amino acid letter and number, and then talk about the type of modifications. So H3K4 ME1 is a single methylation of lysine 4 on histone 3, and then it generally maps to a specific locus on a chromosome. So in terms of getting more specific where you are in terms of the like chromosome 2Q1. The primary histone modifications that are more well studied are acetylation, phosphorylation, methylation, but as I'll talk about later, there's many more different histone modifications, and that can have an influence on the genome as well. So first with histone acetylation, so there's two different enzymatic processes that are responsible for acetylation and then deacetylation. So what it is putting an acetyl group on a lysine, and that changes the charge, and that changes the attraction between those histone proteins and the nucleic acid. And so initially, lysine has a positive charge with the, as seen up here. And with the modification, with the acetylation, which is a covalent modification, it changes it to a neutral charge. And DNA is a slightly negative charge, so that weakens the interaction. So that's how it results in kind of opening up the genome for transcription and stuff. It does occur on both histone tails and the core. Generally, the histone tails are the more, at the site that there is more acetylation and other histone modification activity. Acetyl-CoA is a cofactor for this, and like I said, it's generally associated with the active state and increased gene expression, but epigenetics, nothing is absolute. You definitely have points where there might be acetylation where there's gene silencing as well. So phosphorylation occurs on one of three different amino acids, the serine, the threonine, and tyrosine. It occurs on the N-terminal histone tail itself, and that's controlled by either kinases or phosphatases, and this adds a negative histone charge to the histone. So that negative charge on the histone and negative DNA will detract and open up, and is generally associated with an active state with increased gene expression. And then methylation is a process that doesn't alter the histone charge and can have variable different effects on the DNA and histone protein interactions. And these are just a list of all the other many different histone modifications. So there's many different things you can do. I think the thing to understand about this is that all these modifications generally will affect the interaction between the histone proteins, the nucleosome, the DNA, and then affect the, and that's how one of the ways it's thought to affect transcription and gene expression.
On that last slide, can you explain what some of those things are? And I have no idea, like ubiquitalization. What is that?
So,
And the next one is sumo wrestlers getting it.
I honestly have not memorized all of these. These are just different modifications that, like with methylation, adding a methyl group. So it's changing the different, like adding a carbon group, an amine group, different things like that. So.
Well, just the one. What is ubiquitalization?
I actually don't know off
okay.
the top of my head.
Okay, is there any relationship, like when histone's deacetylated, does it affect DNA, methylation, in other words? Do
Yes,
things move in the same direction, so to speak, or are they totally independent?
They're not independent. It's all related. So I think in areas of deacetylation, there is
More methylation.
or more acetylation. It's never in isolation. It's all kind of happening together. So the next portion of this is the talking about the long and short non-coding RNAs. So this is generally associated with gene silencing. There's multiple different types. So there's microRNA and short interfering RNAs. So those are smaller RNAs that, and then the microRNAs are like around 21, 23 base pair long. And those will either cause degradation of mRNA, or they can cause interact with the complex to cause transcriptional repression. So just stopping translation, so not transcriptional, translational repression. So it can work on both transcription and translation. And like I said, it's generally associated with gene silencing. So this also kind of interacts with the other epigenetic mechanisms I talked about already. So histone modifications, DNA methylation, formation of heterochromatin, and gene silencing. So this generally mainly has a role in gene silencing. So this is a big diagram of kind of the overall steps of microRNA formation and action. So I'll start by talking about initially there's transcription from these transcription factors in the nucleus that causes the formation of this microRNA. So, and it has this hairpin loop structure that gets modified outside the nucleus. So this protein exports the microRNA. There's these different modifications, and it eventually becomes a 22 base pair microRNA that in eukaryotes and animals generally interacts with these other proteins to cause either a pause or halt of translation. I was reading in plants, these generally, and these base pairs don't exactly match the base pairs of the mRNA. So it's kind of versus in plants, what's seen is that the microRNA binds exactly to the strand of mRNA, but generally doesn't interact with this protein complex, and that causes the degradation of the mRNA. So it's a process that's a little different in different organisms. And this is a really big diagram to just kind of show that the role of microRNAs in T-cell differentiation. So generally the take-home message is that T-cells differentiate here from a naive CD4 T-cell into many different types. So these microRNAs allow kind of the fine-tuning of more of a Th1 response versus a Th2 response, so that to help silence other inflammatory genes.
What's the last T-cell set down there? I didn't know what that one was. TFH?
Yeah, it's another type of helper T-cells.
T-follicular.
Yeah.
What?
Follicular is what the F stands for.
How come we don't talk about it much?
Next year, you'll assign it to somebody.
You. Is it an important T-cell subset we should know about?
Yeah.
Yeah, I mean, a lot of people talk about T-follicular helper cells are, involved there in lymph nodes, and I don't know anything specific to say about them. But
Young people are making it too hard.
Yeah, I didn't spend too much time looking that up myself either. So.
But, I mean,
Yeah.
just the more fundamental thing, it's hard enough to know the stats and such that are important in regulation, but you're saying beyond that, there's microRNAs that
Oh yeah, and it's not
drive the development of these subsets?
it's not just the microRNAs. There's so many other epigenetic mechanisms. So I guess my point is that it's a very hard field because, as we'll talk about later, in terms of targeting, we don't have good ways to target a specific epigenetic mechanism. They're general, there's some therapies that inhibit some of the methylation or acetylation enzymes, but that will cause just general, it's not specific yet. So there's just so much that we have to learn, and in terms of, and we don't understand all these interactions either. A lot of the studies I was looking at in terms of asthma and allergic disease and epigenetics is a lot of just observational things, and they're just making conclusions based on things that kind of make sense, but it's still, a lot of it's in mouse models and things like that, and not really kind of at the forefront of clinical application yet. And then the last thing is, the major mechanism is chromatin remodeling. So that's a mechanism by which, using histone modifications in addition to other proteins that kind of come in and change around the histone protein interaction to DNA, and will change the location of nucleosomes and things like that. So one of them is different ATP dependent. There are pretty major modifications that require energy, these remodeling complexes. So it's kind of doing what the histones do on a larger scale. And then just a little bit about one of the newer techniques to kind of start to study and look at all of this. So it's called chromatin immunoprecipitation sequencing. So as we talk about kind of gene sequencing and things like that, looking at epigenetics to kind of start to understand that, this is one of the mechanisms. So for example, trying to go through this, what generally happens is they take the genetic material, cross-link it to, and then select a antibody that binds to a protein and DNA complex, and then so what happens is you're breaking up all the genetic material, and then you're kind of keeping the ones that binds to a specific protein on the histone or something like that, and then sequence all of that genetic material to kind of get an idea of where are all these, you can do histone, histone modifications, or where is that associated? So you're sequencing the gene, and then you're tagging the known protein to try to get an idea of where this is occurring. So kind of getting into the last part of my talk in terms of more clinical application of why all of this is important and what all of this starts to mean. It's looking at one of the things that I found some information about is the relationship of asthma and in-utero exposure, specifically maternal tobacco use, diet, microbial exposures, as I explained earlier, and some of the environmental pollutants. So maternal tobacco use, it has been shown to alter methylation patterns, and there have been some studies that talk about these methylation patterns that are different than, they've looked at methylation patterns in infants who, of mothers who have smoked tobacco, and seen that they're different, and followed them in throughout childhood and seen the persistence of these different methylation patterns. Specific, I don't think we know quite, how to use that information and what that means, but it kind of helps show us that there are non-genetic effects of these of the environment, and it probably explains some of our clinical disease. Another one is a modern diet has that is high in folic acid, which is a methyl donor. So, like a high folate diet, they've studied in some mice that can silence some protective genes for atopy, and have seen that in the mouse model, but that's not something that's really been brought out into clinical application as of yet that I saw. And then they also have been looking at other microbial environments and non-pathogenic bacteria that can induce different epigenetics changes as well. And then lastly, like environmental pollutants as well, has been shown to alter the inflammatory cascade to create more of a pro-inflammatory remodeling of the gene and gene expression through different histone modifications and things like that.
You mentioned folate, a B vitamin. Anybody implicated vitamin D, since that's the trendy vitamin now, and maybe actually will hold up as beneficial?
It's definitely in the literature. I don't. It definitely has been studied. I don't remember specific information regarding vitamin D, but yeah, the information's out there.
As having a role in epigenetics.
And this is just kind of another diagram talking about all those, both, it's not just postnatal environment, but it's that prenatal environment of the diet, microbial exposure, smoking, things like that, in addition to the then changes once after birth as well. And then looking at kind of the expression of the Th1 and Th2 genes, both in utero and postnatally. So epigenetics plays a role in the activation and silencing of Th1 and Th2 genes. So generally in utero, there's an increased Th2 activation that postnatally becomes silenced. And some of the thought is that changes in the environmental changes both in utero and postnatally will kind of alter that balance, and that's mediated through epigenetics. So histone modifications, methylation being some of the more studied ones. So looking at specifically Th1 differentiation, so there's been studies showing methylation of the interferon gamma genes initially, and then demethylation of that gene postnatally, and then how kind of CD4 T helper cells, as they differentiated initially, that it is more, the interferon gamma genes are more hypermethylated, and it undergoes demethylation as it becomes more committed towards the Th1 lineage. So some of those mechanisms are mediated through epigenetics as well. And then environmental factors can definitely alter the methylation pattern. So sometimes, possibly push it towards a Th2 phenotype, and that may play a role in some of the increased atypy and allergy that we're seeing in the last few decades. And then looking at specifically Th2, GATA3 is one of the transcription factors, and this acts at the cytokine locus that, and using histone modifications, there's expression of the IL-4, IL-5, IL-13, and Rad50 is a DNA repair protein, but other Th2 inflammatory protein. And there's methylation of the interferon gamma gene in Th2 helper cells. And then kind of putting that all into context in terms of asthma and epigenetics. So there's been, it kind of is explained a little bit through the Th2 lineage about hypermethylation of the interferon gamma genes and decreased methylation of the IL-4 genes. That affects kind of all of this is mediated through epigenetic mechanisms as playing a role in gene expression. So, if there's external things that affects these epigenetic mechanisms, can start to explain some of what we're seeing. And then there's one of the studies that I'm going to talk about next was done in 2004, and they looked at alveolar macrophages in patients with asthma versus those without. And they, so here I'll show the things that kind of struck me was looking at the different types of enzyme activity between the histone acetyltransferases and the histone deacetylases. So looking at, they found differences in this enzyme activity in terms of in the cells, the macrophages in the airway, but they did not see, this information's not in there, but they didn't see differences in the peripheral blood cells. So it was more in the airway epithelium. So the numbers are small, but what they looked at was more the looking at the what struck me was the treatment with the inhaled corticosteroid would alter this enzyme activity. So is some of the asthma therapy that we're using to like this is the normal control pushing patients and the deacetylases and the acetylases that modifies kind of is used in gene expression of these inflammatory genes that and cytokines in these T cells. So does the inhaled corticosteroid therapy change some of that enzyme activity back to the control, like as seen here in terms of the HDAC and the HAT? So asthma, and then if you add on a inhaled corticosteroid, it pushes this enzyme activity down and brings this one up. So I think some of the therapies we're doing already has epigenetic changes that they're not just that kind of explains a response to therapy and stuff. So better understanding that might be able to help us select out better therapies for our patients.
Since viral illnesses are one of the very predominant things, clearly, that push asthma, and I think actually push the development in genetically predisposed people to autoimmune disease as well, is there a literature on all of that, viral illness and epigenetics?
I'm sure there is. Epigenetics is everywhere. So, I think how different pathogens and microbes act through epigenetics. I think, I don't know if that information's out there. I'm sure it all plays a role and influences, and we're just starting to understand more and more kind of these effects. And then they also looked at looking at gene expression and saw differences in IL-13 and FOXP3 acetylation in patients with asthma as well. So this is just another study looking at 18 healthy controls compared to 14 children with asthma. And I think here, so looking at the Th2 locus, so they found a difference here between an IL-13, and they found a difference in the FOXP3, but it was not statistically significant. So, I think just in terms of they're finding all these associations, and kind of the true relevance of that is how significant are these findings? We're still starting to understand that. And then another study I ran across looked at DNA methylation patterns in children with asthma in the inner city, and found just kind of the overall take-home point of this was there's differential methylated genes with higher and lower. So I saw differentially methylated, so some more methylated regions and some less methylated regions. And how does that all interact? How is that all meaningful? And I think that's, we're starting to, like I said, see these associations. And, I don't think I have quite an answer of how to use this information yet, but...
You remember, is that all in purified T cells that they're looking at?
I don't, yeah. And then just corticosteroids do also, both inhaled and systemic corticosteroids, they do act through different epigenetic mechanisms as well. And then looking at exposure to tobacco smoke, so Bill actually sent out a paper earlier this year looking at increases in lung cancer and oral cancer, lung adenocarcinoma and oral cancer, and looking at these patients' methylation patterns. So some of the cancer biology is mediated through epigenetics, and that kind of that's why epigenetics, especially for tumor biology and stuff, is starting to be more of a target for certain therapies. And then I talked about this earlier, but the prenatal in-utero exposure to tobacco, that they've seen that these alterations in the methylation patterns can persist into adolescence. And how is all that important? Kind of, it just leads to more and more questions, and just shows that how, in its infancy, epigenetics is. So I was going to end with just a little bit about epigenetic therapeutics and what is out there. So the epigenetics, more of the therapeutic information is there for cancer. So specifically, the DNA methyltransferase inhibitor is approved for patients with myelodysplastic syndrome and AML. So like I showed earlier, it kind of alters the balance and changes the DNA methyltransferase in those hematopoietic cells. So that's kind of the thought in terms of using these therapies. And then there's another histone deacetylase inhibitor that's been approved for use in cutaneous T-cell lymphoma. But that's kind of the extent of what I found in terms of epigenetic therapeutic information that's out there. The rest of it is really just in experimental stages. They've tried studies in asthma using this antifungal trichostatin A, and it's a histone deacetylase inhibitor. And I was looking at different papers, and some found increase in asthma, and some found it definitely changes things, and it's definitely. It wasn't a clear-cut thing in terms of would it be beneficial or not, and it's just been studied in the mouse models. So,
like in the cancer drugs, it's a pretty broad hammer to hit it with. Is the idea just you're pushing it away from being a stem cell, and you're trying to terminally differentiate the cancer cells so they can be killed by chemotherapeutics? Or?
Yeah, I believe so. But the thing is, like you said, it's just all of these therapies are so broad. So, what are your effects of you're targeting the specific cancer cells, but you're actually going to hit all your other cells too. So I haven't looked in terms of kind of those side effects of those medications. Could that be more detrimental than the therapy? And I think these ones are they're in phase 3. These two medications, so. It's altering that balance between the stem cells and the terminally differentiating cells. And the last one is the bromodomain inhibitors. So those are also they're looking at ideas of, and the bromodomain is a chromatin remodeling complex, and trying to target this as a possible therapy for multiple sclerosis and cancer. But that's definitely not ready for the clinical stage yet either.
Yeah, as you pointed out already, actually, steroids also work in
Epigenetically.
We don't think of them that way,
yeah.
but I think some data that shows that.
But is that as important as the direct effect, though?
That's a great question. I don't think we know. Or the cause and effect. What came? Do we change epigenetics and that results in a change, or are the changes associated with changes in epigenetics and kind of going forwards and backwards? And I think that's also still not clearly understood.
Well, excellent talk.
Yeah.
Very loud and well spoken. For the future speakers, this is a good example of no one complained to me about not being able to hear. So Nick did this topic how many years ago?
About 12 years ago.
Yeah, so it's come a long way.
Yeah.
Discussion.
We have one more presentation before the holiday break.